Hospitals are an essential part of our health system. They're a place we go to when we need specialised medical care, where we expect to feel safe, cared for and protected. But it's also a place where we can pick up a serious infection. In fact, 1 in 10 patients develop an infection during their stay in hospital. We call this a hospital-acquired infection. Every year, between 165,000 to 200,000 people obtain an infection as a result of their stay in an acute healthcare facility in Australia. Urinary tract infections, surgical site infections, respiratory and bloodstream infections are some of the most common types of hospital-acquired infections. They may be serious or even life-threatening. These infectious organisms may also spread easily in the healthcare environment and lead to outbreaks. Hospital-acquired infections cause unnecessary pain and suffering for patients and their families. In some cases, it can even cause death. A patient's risk of death is about 3 times higher if they acquire an infection in hospital. Also, a patient's length of stay in hospital is increased. They may require additional treatment, investigations, and even surgery, which further burdens our healthcare system. So how do infections spread? In healthcare settings, bacteria are primarily spread through touch or via contact with the surrounding environment. To minimise the risk and spread of infection, healthcare workers maintain high infection control standards. Regular hand washing, maintaining a clean environment, separating infectious patients from others, usually by putting them in a single room, wearing protective equipment like gloves, masks and eye shields, disinfecting medical equipment after use, handling and disposing of sharps appropriately. But sometimes standard precautions are not sufficient, so transmission-based precautions are introduced. Transmission-based precautions are extra steps to prevent the transmission of infectious agents. This includes multi-resistant organisms, bacteria that are resistant to multiple antibiotics. Antimicrobial resistance is now a worldwide problem. Many hospital-acquired infections are caused by bacteria resistant to first-line antibiotics, making treatment even more challenging, especially when results of drug susceptibility testing can take several days. This means we may not know that the bacteria are resistant to the treatment selected. As you can imagine, the longer patients get ineffective treatment, the greater the risk of a poor outcome. Multi-resistant organisms can spread quickly, and 1 in 10 hospitalised adults become colonised or infected with these organisms during their admission. This is especially true for our most vulnerable patients with weakened immune systems from cancer, chemotherapy or transplantation, or those undergoing complex surgical interventions or intensive care. With the threat of drug-resistant bacteria escalating, surveillance and prevention is more important than ever. Detecting outbreaks and identifying how bacteria are spreading is central to effectively managing an outbreak. Traditionally, detecting outbreaks involves looking for a cluster of the same species of bacteria. However, existing laboratory methods have major limitations. Outbreaks can be incorrectly identified when bacteria appear superficially the same. Conventional molecular methods used to type bacteria only analyse fragments of the whole genome, meaning the level of resolution is limited. Strains cultured from a series of patients may be classified as the same genotype by conventional methods. However, when the whole genomes are compared, these strains may be only distantly related, making transmission between these patients unlikely. Scientific advances in sequencing technologies can now assist hospital infection control units to improve infection control responses within hospital and healthcare facilities. Whole genome sequencing helps identify an outbreak, pinpoint the source, and reconstruct transmission pathways. So how does whole genome sequencing work? It is when all the genetic material of the organism is sequenced, not just a few genes. All living organisms are made up of cells that contain DNA, which act as biological instructions on how to develop, function, and reproduce. Any DNA sequence is made up of a long series of molecules called nucleotides. These represent the genetic code which determine the protein structures that allow the cell to function. Over time, changes in this nucleotide sequence accumulate. We call these changes mutations. This may be accelerated in response to antibiotic exposure or following adaptation to the human immune system. Bacterial genomes are also highly adaptable and may acquire new genes from other bacteria or other mobile genetic elements that alter the way genes are expressed. Each bacterial cell's DNA is unique, and this can be revealed by using genomics. By sequencing bacterial genomes, infection control teams can use the genomic information to guide their practice, understand outbreaks when they occur, and identify more problematic strains. The genome sequencing process happens after bacteria are identified from a patient or an environmental sample that are sent to the lab for testing. Detailed genomic information can tell us the differences between strains, even closely related ones. This is done by looking at variations in individual nucleotides within the DNA sequence, so-called single nucleotide polymorphism or SNPs, SNPs. The pattern of SNPs are specific to that organism and its immediate offspring. So we can reconstruct in exquisite detail the evolutionary relationships of different bacteria and how they are related, similar to drawing a family tree. And these relationships reflect transmission events between patients or the environment and help us understand how the spread has occurred. It can also help us to better assess the level of risk a bacterial strain carries. For instance, we can rapidly compare any new strain to previous outbreaks or any sequenced isolate from the international databases. We can also characterise any genes that make it resistant to some antibiotics and more difficult to treat, or virulence factors that make any infection more severe. Whole genome sequencing is also a powerful tool to help infection control staff understand how an infection has spread. This means we can implement the most effective preventive intervention strategies. For example, we may sequence a number of strains from patients on a ward who are all suffering infections with multi-drug resistant bacteria. The genome sequences may show these strains are all closely related, but not identical, with the pattern of SNPs more suggestive of a common environmental source than direct patient-to-patient transmission. This can then prompt additional sampling from other sources, such as sinks, drains, surfaces, or medical equipment. If any strains are identified in these cultures, whole genome sequencing can be used to link these to the clinical cases. If they are found to be identical or near ancestors of the clinical strains, then the likely source of the problem has been identified. This can prompt appropriate responses, such as additional cleaning, decommissioning, removal of contaminated or other engineering solutions.